Hey, it's Jeff Hansen again. One reason I'm making this video is because I hope to review the exam on Tuesday, so we only have so much time to look at carbohydrates and of how your body oxidizes the fuel carbohydrates or lipids in order to make energy. And when I say energy, I mean ATP. So this will be due on Monday night at midnight. And I want to begin in thinking about carbohydrates to classify them. Three really important biomolecules are carbohydrates, amino acids, and hydrocarbons. Will you please describe each of them and compare and contrast them on your worksheet? This should be straightforward. We've talked about it before. Can you label the general category represented by an example from each of these figures? Please pause and do so, but we're going to move on as we set the context. An amino acid has an amine plus an acid linked to the same central carbon, which is alpha, in comparison to the carboxylic acid. The number 1, 2, 3 carbon, the alpha carbon, has 4 different groups coming off it. You should know its stereochemistry. Carbohydrate. Carbohydrate has carbon, represented in green, oxygen represented in red, and hydrogen when linked to an oxygen shown in red, and when linked to a carbon not shown. Hydrocarbon, well, hydrogen and carbon, and that's mostly hydrogen and carbons here. Fatty acids do have some oxygen atoms, but there's very few in comparison to amino acids or carbohydrates. You should be able to interpret these syllables when they occur in chemical names such as ill, ate, ite, or ide to get an insight into what's happening. And of course, I'm using the standard representation where every end of a line represents a carbon atom, every angle represents a carbon atom, and every intersection represents a carbon atom. Now, of course, if a line ends with a letter like O for oxygen, well, there's not a carbon between there. There's only a carbon at the intersection, directly linked to an oxygen and an oxygen. The end of this line, of course, is linked to a nitrogen, so it's a carbon-nitrogen bond, whereas this is a carbon-to-carbon bond. And as I mentioned, there should be how many hydrogen atoms at the end of this fatty acid when you just show one bond to a carbon? How about this atom? Well, the octet rule means you have 4 pairs of electrons. So if you're at the terminus of a line, you'll have 3 hydrogens attached, plus the carbon-to-carbon bonds. That's 4 bonds to carbon. Carbon atoms are almost always with a tetrahedral geometry being linked to nearby atoms. That's if they're in single bonds. If there's a double bond, well then they could be in a simple planar trigonal structure. And of course, if there's an angle in here, that means that this carbon has 1 bond to carbon here, 1 bond to carbon here. You'd have to add 2 hydrogen atoms to make up the octet rule. So let's look again at carbohydrates. Carbon, hydrogen, and oxygen. Hydrogen is water source, or it could be generated from water. And carbohydrates could be seen carbon, hydrogen, oxygen, or it could just be seen as carbon, water, because the atomic constituents of a carbohydrate are such that the number of carbon atoms is approximately equal to the number of oxygen atoms, and the number of hydrogen atoms are approximately twice as many as the carbon. So you go CnH2ON, even though it's not real water molecules. But before going on, I would like to categorize general types of carbohydrates. For example, 3 important categories are there are sugars, starches, and cellulose. Can you look at these and tell me which category is which? Go ahead and label each to your best of your knowledge. In addition, some are called monosaccharides and others are called disaccharides. Will you go ahead and label that? Please do so. But we need to move on. You wouldn't necessarily know this, but I think some of you will. Well, sugars really are derived from the word for sweet. So They need to be small enough to bind to your taste receptors. Hence, we're talking about these relatively small sugars where you have a single sugar or a couple sugars linked together. Starches are polymeric forms of sugar, and in particular, they're polymeric forms of glucose, and they're linked together in a way that they cannot fold tightly, which is really nice because they're these big blobs that are in your liver or in your muscle cells. And since they're so irregularly in shape, they're highly hydrated. There's a lot of water all around them, which means enzymes can grab hold of them and either make them bigger when you need to store more fuel, or they can tear them down when you need to use fuel. In contrast, cellulose is also made by glucose, but it's made with a different type of glycosidic link, which allows it to form extremely straight molecules. And if you have a bunch of kinks and bends in something, it's going to be hydrated in water. It can't fold tightly together. Whereas if you have extremely straight lines, they can bind together extremely tightly. In fact, this material, cellulose, is a major component of tree trunks. It gives amazing strength to a lot of plants. So we have sugars, starches. Starches are the plant form of storage of sugar as a polymer, and glycogen is extremely similar to a starch in terms of the links and all, but it is the animal form of storage for starch in huge polymers. Among the monomeric sugars, the most important for you to know are glucose and fructose, and if you link glucose with fructose, you get sucrose. Alternatively, glucose can be linked to a different monomeric form of sugar to make the disaccharide lactose. Where do you suppose you find lactose? Etymologically thinking, lact? How about lactation? This is milk sugar. Where do you think you find a lot of fructose? Not a surprise. Fructose, a lot of it found in fruit. All of these sugars are found in many different types of organisms, but fructose was named because it's prevalent in fruit. Lactose was named because it's a dimeric sugar, a disaccharide, commonly found in milk. If you hydrolyze the glycosidic link in lactose, you will get a glucose back and a galactose. Anyone know what you call the galaxy? The galaxy? How about the Milky Way? That's mythology and all. And maltose, not as trivial, but malt is basically a type of corn mush that ultimately, they can make it alcoholic and stuff, but you do get a malt, a type of sugar, a disaccharide of glucose in there. So we're going to look at the polymeric forms later, but let's look a little first at the monomeric forms. This giant dendrogram you absolutely do not want to memorize. It's kind of interesting. What do I want you to know from here? Well, I think you should be able to interpret triose, tetrose, pentose, and hexose. Also, what's relevant about all these sugars, I'll illustrate with the hexoses, that they have a lot of chiral centers. At one end, you have the anomeric carbon with a double bond, and since it's anomeric with a double bond, it cannot be linked through 4 different types of bonds, so it is achiral or anomeric. And the carbon at the opposite end is also not chiral because it has 2 hydrogen atoms. And a carbon center has to be linked to 4 different types of atoms or groups in order to be chiral. That means of the 6 carbon atoms in these hexoses, 4 of them will be chiral. So it's interesting to know that of these hexoses, there's going to be a number of types, turns out to be 16, but of those, there's only a few that are really interesting. As I mentioned, glucose, fructose, and galactose. Now it turns out that of a pentoses, that ribose is important because we have deoxyribose in all those coenzymes, coenzyme A, and ADP, FAD, and also it turns out that glyceraldehyde is important. This 3-carbon sugar, one of the simplest ones, glyceraldehyde is important because it's an intermediate in glycolysis. When you burn glucose, when you oxidize it to get energy to make ATP, the glucose gets cleaved into two trioses, one of which has a glyceraldehyde component. And then it undergoes different types of phosphorylation reactions, so it is interesting to be familiar with that. Also, it's interesting to be familiar with dihydroxyacetone. And again, these Fisher diagrams are great. I would rather use these conformational diagrams, and you should be able to interpret the language for each of these. For example, here are the same compounds. Will you go ahead and name them? If we start here, this is 3 carbon atoms and 3 oxygen atoms and however many hydrogen atoms you need to fill it. Glycerol. And if I ask about this structure on the next test, I want you to be able to recognize it by this name, glycerol. Glycer means sweet because this looks a little like part of glucose, so it can bind to part of the taste receptor and signal sweetness, hence sweet alcohol. Name it. Well, what type of group? Aldehyde, formed by the oxidation of glycerol, glycerol, or glyceraldehyde if you want. Ooh, oxidize it again, what do you get? Well, what's the group? Please answer. This is a carboxylic acid made by oxidizing glycerol one more time, hence glycerate or glyceric acid. And now the last one, interestingly, is similar to B. You've singly oxidized glycerol, but now you've done it on the central carbon. What do you call a carbonyl carbon? That's a carbon-oxygen double bond with a carbon on each side of it. You call it a ketone. Hence, it's called acetone. It's a ketone that's really similar to acetic acid, but it has an extra carbon to get the ketone, and it's called dihydroxy. Why dihydroxy? Because there are 2 hydroxy groups on it. Kind of a complicated name, but it comes out of the organic nomenclature. And here's what I just described. And I want to remind you that the final is comprehensive. You can't really understand what I've just described unless you kind of know what an acetyl group is, acetate, hydroxyl, aldehyde, ketone, carboxylic acid. So I can ask about any of these earlier learning objectives. I may not just ask about them, I might ask you to apply them to what we're learning about sugar metabolism. And likewise, we still need to know Arabic numerals and Greek letter nomenclature. You can't just memorize this, you need to think it's so logical the way it's done. And again, we're going to use a lot of nucleophilic attack steps in looking at sugars. In addition, there's going to be a little bit of new nomenclature. We did look at the anomeric carbon before. I'm going to define that more carefully again. And again, notice glucose, linear glucose, alpha glucose, beta glucose, fructose, sucrose, galactose, lactose, aldose, ketose, starch, amylose, amylopectin, cellulose, table sugar, and high fructose corn syrup. Awful lot of oses in there because they're sugars. This type of nomenclature underlies our discussion of carbohydrates. And I'm going to ask you to compare and contrast some different types of sugars. But I want to begin with a question. Both potatoes and table sugar are made pretty much out of sugar. In the case of a potato, it's a polymeric form of sugar. In the case of table sugar, it's a dimeric form of sugar, a disaccharide. Whether you eat a potato or table sugar, they are very rapidly hydrolyzed down to monomers, which are very rapidly absorbed by your digestive system. So, nutritionally, are they equivalent? Please pause and explain. And I'm going to say, my answer is absolutely not. They are extremely different because the potato is made out of starch, which is pure glucose. Starches are half of our dietary energy, whether it's in the form of wheat or corn or rice or potatoes, is pure glucose, but in a polymeric form. In contrast, table sugar is a dimer, but it's not made of pure glucose like potatoes. Rather, it's a dimer between glucose and fructose. Therefore, if you eat a baked potato, that starch will rapidly be digested, and in your intestine, you will have a massive amount of only glucose monomers. However, If you take in the disaccharide of table sugar, it also is digested very rapidly, and in your intestine you will have a very different mixture. Half of it will be the same glucose as you get with a potato, but importantly, the other half of this will be fructose, which is actually harmful. In the case of the potato, as it gets completely digested down to solely glucose, your intestine absorbs it, it gets into your blood. Insulin and glucagon regulate the uptake of glucose into cells, whereas insulin and glucagon don't regulate fructose. Inside of the cell, glucose metabolism is carefully regulated between the different needs of the cells. However, fructose is not regulated among the different needs of the cell, which can contribute to metabolic diseases such as non-alcoholic fatty liver disease. Accordingly, I want to look at these two really important monosaccharides, glucose and fructose, and I want to look at the dimer, sucrose, and how it gets hydrolyzed by a water molecule to release them. For example, first of all, what would you name the enzyme that hydrolyzes the disaccharide sucrose into the two monosaccharides, glucose and fructose? That much nomenclature, it's worth memorizing, and you're going to build from this type of nomenclature to the higher levels of complexity in terms of pathways and diseases. What do you call the enzyme? You could have said many things and you wouldn't be wrong. Technically, the name is sucrase. If you said, let's see, it acts on sucrose, so the first name is sucrose substrate. What does it do? It hydrolyzes it. How about sucrose hydrolase? Perfectly good name. And you don't have to worry. If that's what you came up with, I'm pretty happy, because sucrase hydrolase could have been the name of it, and therefore I think you'll be able to interpret sucrase. A is the enzyme that acts upon sucrose. So interpretation is where it's at, not memorization. What other structural component of glucose and fructose do we need to understand? At the heart of these sugars is the fact that they are in equilibrium. Glucose is in equilibrium between a cyclic and linear form, and fructose exists in equilibrium between a cyclic and linear form. That you have to know cold. Also, you need to know which is a ketose and which is an aldose. Please pause and tell me. Again, basic organic nomenclature. Ketose, os always means sugar. Ket is from ketone. Aldose, os is from sugar. Al is from aldehyde. Which is an aldehyde, which is a ketone? Aldehydes and ketones are based upon a carbonyl group. A carbonyl being a carbon-oxygen double bond. What distinguishes them is that in the aldehyde, the carbonyl carbon is at the end of a carbon chain, such that adjacent to it will be a small hydrogen atom. In contrast, in a ketone, the carbonyl is not at the end of the carbon chain. Rather, there's a carbon atom adjacent to the carbonyl on both sides. How will that affect the reactivity of aldehydes versus ketones? Please answer. Do not memorize one is more reactive than the other. Think about the logic behind it. Well, when we're talking about reactions, what is the mechanism we're always thinking of? A nucleophilic attack. So a nucleophile will need to get access to the carbonyl carbon. How will it do it? In the case of an aldehyde, there is a big carbon chain that will get in the way somewhat, but on one side of the aldehyde is a lone little bitty hydrogen atom, so it's much easier for a nucleophile to get access to the carbonyl in an aldehyde than in a ketone. In a ketone, the carbonyl carbon is surrounded on both sides by large carbon atoms, on one side an entire chain, on the other side at least one carbon with oxygen and hydrogen atoms. Therefore, it is harder for the nucleophile to get into that narrow opening in order to attack the carbonyl carbon. At this point, I'm sure some of you are saying, I don't quite get the chemistry, so I'll just memorize that an aldehyde is more reactive than a ketone. That would not be a good idea. Certainly you can use brute force memorization to learn almost anything short term. I'm quite sure most of you will suffer some type of cognitive overload because some things you just do have to memorize. You can reduce that burden greatly by not using brute force memorization when you have the alternative of understanding the concept, especially when it is so broadly applicable. Why is this broadly applicable? Why do you think the FDA says you shouldn't drink formaldehyde? It is that extreme reactivity of the aldehyde group that's so toxic throughout your body. And for that matter, how about the aldehyde group on glucose? It is that reactive aldehyde group on glucose which makes the hyperglycemia so harmful in chronic diabetes. And to understand that chemistry, as well as the nomenclature of the enzymes and the products and substrates of the metabolism of glucose, it helps to know the numbering of the carbons. WU applies the rules of nomenclature to number each of the carbon atoms in linear glucose and in linear fructose. Please pause and do that. Let's look at glucose first. Unlike a fatty acid that only has oxygen atoms attached to one carbon, here we have an oxygen linked to every carbon atom. Therefore, we need to find something unique about one of the atoms in order to find a landmark. What is that? I think most of you know. If you don't immediately know, take a look and see if you can figure it out. And the answer is that, oh, one of the oxygen atoms is different. It's in a double bond. And two bonds to the heaviest atom is more important than one bond to the heaviest atom. So in sugars, it is that carbon atom that's bonded twice to oxygen that's important, and it is extremely important. This is probably the most important learning objective in the entire presentation. This is called the anomeric carbon. If you can't find the anomeric carbon in a sugar, you're wasting your time. And the principle is simple. You don't really have to memorize anything because it is clearly distinct from the other oxygen atoms. Okay, so now we have the anomeric carbon. Number all of the carbon atoms in glucose. Well, using the landmark anomeric carbon, it's easy. 1, 2, 3, 4, 5, 6 going to the far end. Now, how about numbering fructose? Again, with any sugar, the most important first step is to identify that anomeric carbon. Please do so. Pretty simple. Here it is, the carbon with a double bond to oxygen. Now can you number? And of course, there's a little bitty trick there, not too bad, but hopefully you got that. You don't start at the anomeric carbon, because then what direction do you do the numbering in? Rather, you start at the end closest to the anomeric carbon and number toward the opposite end. And so accordingly, in glucose, the anomeric carbon is carbon 1, and in fructose, the anomeric carbon is carbon 2. Will you now number the oxygen atoms in the linear form of glucose and fructose? Please pause and do so. And again, I'm trying to show you there's no memorization. The numbering is exactly what you'd expect it to be. How would you logically number the oxygen atoms? I think every one of you would come up with what the organic chemist came up with. You would number the oxygen atom based upon the carbon atom to which it is attached. And again, I want to be clear, please do not even think about trying to memorize these Fisher diagrams with a hydrogen on one side and oxygen on the other. That would be a complete waste of time. We are going to look at the stereochemistry of the individual carbons, but it'll be based on a really trivially simple algorithm based on the conformational figure, not on the Fisher diagrams. What you do need to know about the Fisher diagrams is the anomeric carbon. The anomeric carbon of glucose is carbon 1. The anomeric carbon of fructose is carbon 2, which makes glucose an aldose and very reactive, and fructose a ketose and less reactive. Which of course means that if you're looking at the stereochemistry of glucose and fructose, they're almost identical. It simply has been the movement of the double bond from carbon 1 to carbon 2 that distinguishes these. And so even though glucose and fructose are chemically very similar, the same number of carbon, oxygen, and hydrogen atoms, very similar stereochemistry, When you do move the double bond, you have the same numbering for the carbon and oxygen atoms, equally water soluble, and yet glucose and fructose are profoundly different biologically, especially with regard to human health. But right now, I really want to zoom in on this profoundly important nutrient, glucose. And I've already mentioned how glucose interconverts between a linear and cyclic form. Right now, I would like you to pause the video to describe that reaction. Will you please draw arrows to represent the intramolecular reaction by which linear glucose cyclizes to form cyclic glucose? If you need a clue, it's going to involve the one mechanism I always talk about. There will be what? Yeah, a nucleophilic attack, which should tell you where the attack will occur. Please go ahead and do that. The first thing I'm going to do is I look for that double bond. Where's that important anomeric carbon in the cyclic form? What simplifies this problem is to realize that the anomeric carbon will have two bonds to oxygen, whether it's in the linear or cyclic form. That is, the nucleophilic attack on the carbonyl carbon will cause the electrons to resonate up onto the oxygen atom, and it'll grab a proton, but that means that the linking oxygen and an exocyclic oxygen will both be linked to that carbonyl. So we look at the cyclic form to look for which of the carbon atoms is bonded to two oxygen atoms. Please do that. It's in your interest. It is absolutely in your interest to spend a few minutes here to understand this, and you'll remember this very long term. I look at this carbon here, right? And I go, okay, there's one oxygen atom here. Let's go to the left. Oh, a carbon here. Go to the right. Oh, there's a carbon here. There's a hydrogen atom going straight down from it. Not anomeric. Another clue is that when the oxygen atom nucleophilically attacks the carbonyl, that means that the linking oxygen in the ring is going to be attached to the anomeric carbon. So we know it'll be next to the ring oxygen. We can look at the carbon adjacent to the left, and we can look at the carbon adjacent to the right. The carbon atom on the left is bonded to the ring oxygen, then it's bonded to a carbon atom here, a carbon atom here, a hydrogen below it, only one bond to an oxygen atom. Go to the carbon atom on the right, go straight to an oxygen atom. The anomeric carbon is linked to the ring oxygen and to an exocyclic oxygen atom, so therefore you've identified the carbonyl carbon. So to summarize, in thinking about going from linear to cyclic forms of sugars, first of all, in the linear form you look for a double bond. In the cyclic form you look for two single bonds to oxygen, so that either way the carbonyl carbon has two bonds to oxygen. In addition, by default, the anomeric carbon should be on the right side of a conformational figure. So that's also a clue, but always look for it being next to the ring oxygen and having two bonds to oxygen atoms. And now that we can see how the ring formed, will you number the carbon atoms first in linear glucose and then also in cyclic glucose? Please go ahead and do so. But I need to move on. With linear glucose, that's very easy. We simply look for the carbonyl 1 through 6 to the opposite end, right? Now with cyclic, it's a little more difficult. However, we know that carbon 1 is the anomeric carbon, and if we're looking for a continuous carbon chain, we can't go around the circle on the top here because there's an oxygen atom. The only way you can number is to the left on the bottom here, 2, 3, 4, 5, 6. And again, since the nucleophilic attack on the aldehyde group happened from carbon 5, that means that carbon 6 remains exocyclic. It's not in the ring, but it stands out, which makes that hydroxyl a little different from the other ones. An even more important learning objective is that cyclization of linear glucose leads to two different conformations, alpha glucose and beta glucose. This occurs because the nucleophilic attack of oxygen 5 on the aldehyde group can either occur from above the ring or from below the ring, which will push the hydroxyl group either up above the ring of the glucose or down below the ring of the glucose. And the difference in stereochemistry between alpha and beta glucose plays a really important role that differentiates between half of our dietary energy, digestible starches, and one of our most important dietary fibers, which is indigestible, called cellulose. Accordingly, understanding human nutrition makes more sense if you understand the difference between alpha and beta glucose. For that matter, a number of enzymes which are implicated in metabolic diseases because of the way they interact with carbohydrates are named based upon the alpha and beta nomenclature. Since alpha and beta glucose lead to very different biology, this again is something I want you to memorize. And this is one case where I don't resort to pure logic or pure systematic nomenclature. Rather, I resort to a trick. I'm embarrassed, okay? But we have to do it. The simplest algorithm I know about is to say that alpha is like a fish, and it swims below the plane of the glucose, and beta is like a bird that flies above the plane. And the reason I mention this is I myself have never found a logical way to memorize this, a very simple algorithm that will help you understand the names of a number of different enzymes and how starch works versus how cellulose, the fiber in our diet, works. If you come up with a better algorithm for differentiating between alpha and beta, hey, let me know about it. In the meantime, I recommend that you not only try to differentiate between alpha and beta forms of glucose, but I recommend you build each form on your molecular model, and it'll give you a feel for the difference between whether hydrogen or oxygen are equatorial or whether they are axial. You can continue if you want, but the next part of this is due Wednesday at midnight, and that's going to look much more carefully at both alpha and beta forms of glucose and apply them to different things, especially starch and cellulose. So again, any questions on this, don't hesitate to call me. If you do make a molecular model, I hope you shoot a picture of them and include them with your documentation. Thank you.